into our expert pioneering research capsules, which I'll now start sharing my screen.
Thank you so much, panelists.
Thank you so much to all of our panelists. Our next distinguished speaker is a professor of medicine at the Ivy League Weill Cornell Medicine-Qatar, the assistant dean for clinical investigations, and a consultant endocrinologist at Hamad Medical Corporation. He is an international titan in the study of diabetic neuropathy and systemic neurodegeneration, having authored over 150 peer-reviewed publications, delivered over 500 global presentations, and commanded millions of dollars in research funding from the NIH, JDRF, and the European Union. It is our absolute privilege and honor to welcome distinguished Professor Rayaz Malik for his presentation, "The Eye is the Window to Neurodegenerative Diseases and Beyond." And unfortunately, since he could not be here at this present moment, we will be sharing his talk as a pre-recorded one. Okay, can everyone see the video? Just wait, hold on. One moment. I will turn on the sound so that you can hear his external voice as well.
Okay, right. So I'd like to thank.
I'm just going to pause here just so that can anyone thumbs up if you can hear the audio?
Zara for asking me to speak. And I think the objective is to try and give you an idea of anything is possible, but you have to work hard and you have to believe in yourself. That's the key. And what I'm going to talk about is actually, it really is a journey that I've been on for a long time, but hopefully, inshallah, we've come to something very, very good. And the title of my talk is "The Eye is a Window to Neurodegenerative Diseases." So what are neurodegenerative diseases? Well, first of all, they are extremely common. 3.4 billion people worldwide live with some kind of neurological condition. And that, of course, not only is it highly prevalent, but there's two forms. There's the peripheral nerve damage that you can get, and there's a lot of diseases in the brain, which we're all very familiar with, things like dementia, Parkinson's, multiple sclerosis. Essentially, a long list of neurodegenerative diseases are present, and they affect across the world. And that prevalence is increasing as we age more, as we have more risk factors. So what is the problem with neurodegenerative disease? Well, actually, how did I get involved? Honestly, this was when I was still a student. I was very good at anatomy, and I loved nerves, and I just kind of thought, wow, this is a great area to be in. And I ended up doing some research in diabetic neuropathy. Now, diabetic neuropathy is a condition that affects people with diabetes. And I'll tell you, it has major consequences. But some of the things it does, it gives you pain in your feet at night, you can't sleep. And really, we really had a very, very poor understanding of what causes diabetic neuropathy, but more importantly, how do we measure it very, very early on? So here are the manifestations: pain, particularly at night, but also foot ulcers. Now, what is the consequence of having a foot ulcer? Well, not very good. Basically, this is Mr. von Bergmann in Berlin, 1901. What is he doing? He's actually undertaking an amputation. Why would this person be having amputation? Removing of their leg? Because they developed a foot ulcer, they became infected, and then basically the leg was dead, so they had to remove it. Now, how do you deal with this? Well, you can either wait until the patient develops a foot ulcer, or you actually and why do they develop a foot ulcer? Because they have underlying nerve damage. Unfortunately, a lot of pharmaceutical companies are really, I would say, disheartened, dismayed with diabetic neuropathy. We have no current treatments, and We also have, or did have, a very, I would say, not very easy way of measuring nerve damage, particularly in the early stages. And so this is a quote from a head of late complications for a big pharmaceutical company who I happened to meet at dinner. He said, diabetic neuropathy is a nightmare. You have no clear endpoints. So we focus on nephropathy, which is kidney damage. And in fact, the FDA that develop or allow the approval of new drugs said progress in the development of disease-modifying agents in diabetic neuropathy is completely stalled. And we still, to this day, 2026, have no treatment for diabetic neuropathy. From diabetic neuropathy, I take you to dementia. And actually, Bill and Melinda Gates Foundation, you may have heard of the Gates Foundation. Bill Gates was asked to invest over 100 million into dementia. And he got a lot of clever people to look at what was the fundamental problem with dementia. Well, this is what he worked out. Basically, he said that, look, by the time people come to the doctor with dementia, i.e. memory problems, and they've actually already had the disease for about 20 years. And we've missed the boat. So what we need to do is have better ways of identifying the nerve damage early on. And he said, reliable, affordable, and accessible biomarkers have the potential to revolutionize how we approach Alzheimer's disease. He didn't say we're going to have some amazing new therapy or some amazing new, immune therapy or whatever. No, he said, look, if we can diagnose this condition earlier, we can do something about it. And that's where I am going to tell you what we've been doing which is challenging the dogma on neurodegenerative diseases. That is that most neurodegenerative diseases, unfortunately, we are currently dealing with when it's too late, right? This is the end stage. This is whether it's peripheral neuropathy, whether it's diabetic neuropathy, the nerves are all dying or dead. Even in the brain, a lot of advanced damage is present. And what we need to be doing is actually measuring these nerves when in the very early stages of disease, just when it's not even clinical. And that has been a big, big issue because we don't have any good tests to measure early nerve damage. So in 2001, that's a long time ago, and it shows you a little bit about perseverance as well. I and a few other people had a crazy idea, which was a way to measure early neurodegeneration. So instead of taking the patient at this stage when they've got advanced disease of their nerves, what we thought is, you know what, we'll look at the eye. And why the eye? Well, because the cornea, which is the front of the eye, not the back of the eye, the retina, the cornea, actually, just a fact, is the most densely innervated tissue in the body. And you can tell that by if you just touch the front of your eye, you can see how sensitive it is. But just because there are nerves there, how are we going to look at them? Well, actually, that's where the technique that I'm going to tell you about allows us to, in a non-invasive, rapid way, image the nerve fibers in the cornea. And what is that technique? Well, it's called corneal confocal microscopy. So this is a camera which you touches the front of the cornea, as you can see. And when it does, it images the nerve fibers in that cornea. Can you imagine a cornea is only about maybe 1 millimeter thick, and literally it's able to look at nerve fibers. And Einstein said how we think determines what we measure. And if we keep thinking that we need to measure nerve damage in the brain or in the peripheral nerves, then nothing changes. So we did our very first study. You're talking back in 2003. We took 18 people with diabetes and we scanned their eyes. And what did we find? Amazing. Healthy controls, what you see here are white lines, which are nerve fibers in the cornea. If you progress to people with early subclinical neuropathy, there already is a loss of nerve fibers. And furthermore, in of course, advanced disease, you've got very few fibers. So what you see here is a plot of nerve fiber length, and you can see it progressively reducing with increasing severity of neuropathy. And in particular, you're picking up a disease early, which no current test can pick up. We then have also applied AI, and AI is amazing, honestly. The AI can now look at these images, and we use these deep learning algorithms, so it recognizes what's a nerve, what's not a nerve, and literally it is able to diagnose diabetic neuropathy with 100% sensitivity and 95% specificity. That means virtually you're missing nobody who's got the disease and not picking up people who haven't got the disease. And furthermore, even within diabetic patients, we can differentiate those with and without diabetic neuropathy. So we did diabetic neuropathy, showed that corneal confocal microscopy can pick up nerve damage. And then actually, whilst waiting for the FDA to approve things, we and others have shown that in virtually every other peripheral neuropathy, so these can be, from hereditary changes, from inflammatory changes, from chemotherapy, Fabry's disease, HIV neuropathy, amyloid neuropathy, fibromyalgia, we can actually use corneal confocal microscopy to detect nerve damage in these conditions. And the literature has grown and grown from when we started. There was nothing, absolutely nothing. And now you literally, virtually every peripheral neuropathy you can think of, we've been able to show that CCM detects this. But of course, I know what you're thinking. So if chemotherapy-induced neuropathy's got it, diabetic neuropathy's got it, how do I differentiate what's going on? Well, that's where, and I know what you're thinking, and that is where actually AI comes into this. We've now taken the images from people, healthy controls, people with HIV neuropathy, people with chemotherapy neuropathy, people with diabetic neuropathy, people with inflammatory neuropathy, and we've fed them into the AI. And first of all, if you look at the images, what we see very quickly is that compared to healthy control individuals, actually there is damage, but the damage is pretty comparable to my human eye. But when you use AI and particular vision transformers, these not only look at the amount of nerves there, but they look at just amazing things like texture, the shape, the spatial relationship. And when it does that, it almost gets a fingerprint for that particular disease. And so when we do that, you see that virtually every single disease, so whether it's inflammatory neuropathy, chemotherapy-induced neuropathy, the AI is able to say, yeah, this is absolutely certainly this. And what you then get is 100% sensitivity for chemotherapy neuropathy, 99% for HIV neuropathy, 95% for inflammatory neuropathy, and 88% for diabetic neuropathy. So virtually, I can tell you now whether you've got nerve damage and what disease it's coming from. So then I arrived in Cornell in Qatar in June 2014, and I'm bored. And also I'm thinking, well, you know what? The cornea is pretty near the brain. So let's have a look at maybe some diseases in the brain, because there are nerves in the brain that get damaged. Maybe the nerves in the cornea also get damaged. So we look at things like dementia, Parkinson's, multiple sclerosis, even stroke. And what do we show? Well, our very first study that we did was in people with multiple sclerosis. And we were the first in the world to show this. So generally, if you think about multiple sclerosis, people think it's a disease of the brain and the optic nerves. And what we were able to show is actually, no, corneal nerve damage is also present in multiple sclerosis. And it's more serious, the damage, in those who have progressive multiple sclerosis as opposed to relapsing remitting. And moreover, over time, what we showed is that the ones that are progressing faster actually have greater loss of nerves and relates to brain scan abnormalities. We also have looked at dementia and Parkinson's disease, and what we show there is beautiful, because we show that healthy controls, even in people who've got mild cognitive impairment, which is actually, it's not dementia, right? It's like 15, 20 years before this all happens, there is already a loss of nerve fibers. So in people actually with Parkinson's disease, with cognitively normal, there is less nerves. But in those with mild cognitive impairment, there is even less nerves, and those with dementia, they have the least nerves. And the amount of nerve fibers, it actually relates to the cognitive function. And you get very high sensitivity and specificity, which basically means you're picking up all these patients with dementia. And furthermore, we've also shown that actually you can predict those who develop dementia based on their baseline scans. So the already people who are going to develop dementia have lower counts for nerve fibers. So we did multiple sclerosis, and we've done dementia, Parkinson's disease, motor neuron disease, other people have done. Autism we've done, schizophrenia, even long COVID, epilepsy, migraine, and stroke. And so again, the literature, starting with us, alhamdulillah, has grown and grown and grown. But again, I know what you're thinking. How do you differentiate these different central neurodegenerative diseases? Well, we've done it. We've compared dementia with multiple sclerosis, with Parkinson's disease. Again, you see this loss of nerve fibers uniformly across all three conditions. But again, AI is so clever that it's able to differentiate these diseases with almost 100%. Well, dementia is 93% sensitivity, 96% specificity. MS is 92%, 95%. Parkinson's disease is 100% differentiation. So we have a eye scan that can identify neurodegeneration, but also pick up which disease it's coming from. So the evolution of CCM really starts with a crazy idea in 2001, which we then get a grant in 2002, literally $52,000. And we show that in 2003, that's the key paper. And then actually, honestly, you've got to be thick-skinned, because I remember presenting that paper as a junior doctor and people literally getting up and shouting at me, for senior professors saying, you don't know what you're talking about, you've got it all wrong. And then literally we had a struggle. And I like using this video because it gives you an idea of, even in academia, people are fighting, right? Because we're challenging the dogma, challenging the way people are thinking. You've got to think of it as a word like that. Eventually, we actually have now ended up with a new term called neurooculomics, which is scan the eye and work out what's going on in terms of neurodegenerative diseases. So my future, the way I see it, neurooculomics is going to be people are going to go somewhere. Could be a hospital, could be a clinic, could be an optometrist. They're going to have the scan, CCM. AI is first of all going to tell you whether you're normal. If you're normal, you're healthy, you go away. However, AI will also identify people who are abnormal, maybe in the preclinical phase, maybe in the advanced phase. And then AI will then again differentiate people, whether you've got diabetic neuropathy, you've got Parkinson's disease, or you've got dementia. So what started off as a crazy idea in Manchester has now grown all over the world. And a lot of people are doing this, and hopefully, inshallah, soon, we'll be able to roll this out into the population. And I'll finish by saying the eye is the window to the soul, and not because I can't spell correctly, but soul as in the feet where neuropathy affects, and the brain. Thank you.
Professor Malik. Okay, so we finished with Professor Malik. Thank you so much for that. It was pre-recorded, but still, that was a masterclass in clinical discovery.